Thank you for joining us today for Unlocking Soil Secrets, Advanced Analysis of Soil Surfaces using X-ray Photoelectron Spectroscopy. We are so glad that you're here to join us today. Our next EMSL Learn webinar is going to be on May 28th, and it will focus on how you can use EMSL's vibrational sum frequency spectroscopy to accelerate your biological and environmental research. For more information will be provided for you through our social media channels and via email for all of our upcoming events and other opportunities. So make sure that you keep in touch with EMSL by subscribing to our email and social media channels if you haven't already. Links will be provided for you in the chat. So for today, our speakers include EMSL material scientist Shuttha Shutthanandan, who will provide an overview of X-ray photoelectron spectroscopy. Ooh, that's a mouthful. And it's a widely used surface analysis technique. And he will also explain how EMSL's new XPS, high-throughput soil analyzer, can accelerate your elemental soil interrogations. Our other speaker is EMSL user Catherine Pettinger from the University of Wisconsin-Madison, who will demonstrate how she used the high-throughput soil analyzer for her research. And we also have joining us Ajay Karakoti, a PNNL material scientist, who will discuss how to leverage artificial intelligence and machine learning tools for XPS surface analysis capabilities. There will be time at the end of today's webinar for questions, so we encourage you to post your questions in the Zoom chat so that our speakers can answer them during the Q&A portion after their presentations. So with that, we will begin with Shuttha.
Heather, can you give me the access? It's not. Okay. Thank you, Linda. Thanks for the introduction. So today we are going to show you guys how we can use XPS or X-ray photoelectron spectroscopy in advanced soil analysis. So this project is funded by EMSL dash proposal, FY24 dash proposal. And then here is our team from different people from different directorate. So it is a very successful project. And then we will show you some data from this high-throughput methodology. So first, why do we need a surface-sensitive technique in soil analysis? As we all know, the surface of the soil particles play an important role where most of the reaction actually occurs, such as absorption of pollution, a nutrient exchange, and then microbial interaction as shown in this figure. So these particular microbes or the nutrient going to interact first with this, the mark in the red line here, which is the outermost layer of that soil particle. So understanding the surface chemistry is very critical for predicting how contaminant move through the environment or how nutrient become bioavailable. So to understand this, we need a surface-sensitive technique, and XPS is one of them. So I'm going to show how we use that in this talk. So at first, I'm going to give you some basic principle of XPS, what it can do, and what is the strength of the XPS. And then I will give you a couple of examples on our old work, and then I will go to the high-throughput work, and then Catherine and Ajay will continue. So what is XPS? The XPS is basically you shoot X-rays to the samples. As we all know, in one sample, if you take an atom, the atom have a lot of electrons at different energy level, orbiting different energy levels. And then when the X-rays hit the sample, if you have enough energy, what is going to happen? It is going to kick that electron out. We call that is a photoelectron emission. And then this electron is going to come with the energy of E of case. So before we inject a photo X-rays, the atom will have energy of E sub I. Then, because of we lost one electron, it is going to have energy of E sub F. So using the conservation of energy, we can equate the initial energy, which is h nu, the X-ray energy, plus the atom energy is equal to the kinetic energy of the electrons emitted plus the final state of the energy of the atom. So we can rearrange this equation like this. So that gives you h nu minus Ek is equal to E sub F minus E sub I. Basically, this is a binding energy of an electron, particular electron at a particular atom. So if you know h nu, because we know exactly what X-ray energy we are shooting, and then somehow if you can measure the kinetic energy of the electrons coming out during this process, then using this equation, we can calculate the binding energy of that electron in that particular atom. So that's what it is important, because using this equation, we can identify what are the elements present in any samples, any soil sample. So XPS, we run in basically two different mode. One is called survey scan. Basically, in this scan, we scan the kinetic energy of the electrons coming out from this process, and then we monitor how much the photoelectrons are coming out. So in a fast mode, you can say I'm running in a fast mode. So this is a typical X-ray XPS spectrum from a silicon dioxide sample. You can see there are several features here. One is sharp peaks, and then some continuous background, and some additional extra features. So I will explain one by one. So the first one is called the photoelectron peak, which is the electrons coming out from the sample without losing any energy. When the X-rays are hitting the sample, it is generating the electrons from that particular spot, and then these electrons came out without losing energy. So that is this one. So this one, as I mentioned, it has each has a different binding energy, so you can identify them. And then it is surface sensitive because it can only come from the top surface. The second one is called the background of the spectrum, which is shown here. That comes because assume that the electrons are emitted from deeper into the sample, because X-rays can penetrate deeper into the sample. So when the electrons coming out from the solid, it is going to interact with some of the electrons cloud from the other atoms, or sometime in the nucleus. Because of that interaction, it is going to lose energy. So when it's losing energy, when it comes out from the surface, it is going to have a much different energies, much smaller energy than the actual photoelectron energy. So this is a continuous process. That's the reason we get this background. The third one is actually the relaxation phenomena we call OJ peaks. It is a 2-electron process. I'm not going to go into the detail, but that also useful in some certain condition. So basically, we identify the peak using the binding energy, and then using the area under the curve, what we can do, we can convert this area under the curve using the known photoelectron cross-section and some experimental parameter. We can convert this area into the area under the curve into the quantification. The second mode that we run the XPS is called high resolution mode. Basically, what we do, we pick one photoelectron peak and then scan very slowly using high resolution, high energy resolution, so we can separate some different peaks if any they are in that region. So that high resolution scan will give us some additional information we call chemical state. I will explain what does that mean. We call chemical shape. So basically, the co-level binding energy is determined by what is the electrostatic interaction between the electron and nucleus. So when the X-rays hit, that is the bond that it has to break to remove that electron out. So if you, for example, if you take the carbon has a 1s electron, it also have a 2p electron. So these are co-level, and these are the valence electron, and then it has a nucleus. So this carbon 1s electron is interaction with this valence electron as well as the nucleus. So this valence electron is going to screen some of the interaction happening at here. So what will happen if you bring the oxygen there? So if carbon bonded to oxygen, we know oxygen is the electronegative element, so it is going to attract electron. So this valence electron are attracted towards the oxygen. So that means this interaction is less, or the loss of screening. So this carbon is going to be attracted more towards the nucleus. So now it has a highly tightly bounded to the nucleus. So we need to supply more energy, more X-ray energy to remove this one. this particular electron, that will give you the shift in the binding energy. That's what we call chemical shift. So this is basically true for the carbon compound, the carbon-containing compound. So here is an example of a carbon 1s spectrum from the ethyl trifluoroacetate. So you can see here the carbon has in 4 different chemical environments. Here it is bonded to 1 carbon and 3 hydrogen. Here is bonded to oxygen and hydrogen. Here is bonded to 2 oxygen. Here is bonded to 3 fluorine. So you can see each bonding is giving you a different speed because of the energy of these, the energy of the needed energy to remove the photoelectron is different from these 4 different configurations. So these are the things we call chemical shift. And then you can see these peaks are all same height because we have same amount of carbon present in this particular sample. So this is the main advantage of the XPS, that we can get some chemical information in carbon compound, and we can also get information about the oxide and carbonate and things like that. So the rule of thumb is the larger the electronegativity element is, the larger the peak is going to be. So you can see fluorine has larger electronegativity compared to the oxygen. That's the reason this peak is coming far apart. So here is the data I am showing. This is the summary of the data that we obtained from all kind of literature available in the, literature available for us. And then you can see when carbon bonded to different elements, it has a different binding energy. So by using these tables, that we can easily separate whether we have a CW, the carbonic acid bond, or we have a carbon-carbon, or we can carbon-chlorine and carbon-fluorine, things like that. Okay. So this is the snapshot of the photoelectron spectroscopy. So we can basically measure from lithium to uranium. We can get quantitative analysis. As I mentioned, we can get nice chemical information, chemical state information, and the analysis depth is around 10 nanometer, and the detection limit is around 0.1 atomic percentage. Okay, so now I am going to show you how we can use XPS in the traditional soil analysis. So first one is the carbon speciation. So basically, the carbon speciation is, as I mentioned, here is the carbon 1s spectrum from some soil sample. You can see in order to fit this carbon 1s spectrum, I had to fit with 5 different peaks here. So these 5 different peaks, we didn't choose arbitrarily because we chose this one based on these binding energies. These binding energies are corresponding to this functional group, and these functional groups are actually connected to these metabolites. So if you have a carbon-carbon-hydrogen bond, that belongs to lipid and lignin and things like that. So this is in corresponding to the different oxidation state of carbon. So by doing this kind of a deconvolution of this main peak, we can get more information of what kind of carbon speciation is happening in the soil sample surfaces. We can also do the similar thing for the nitrogen. So here's again nitrogen spectrum from a soil sample. Here we have to fit with 2 peaks. One peak, the main peak, is coming from the amine group, and the green curve is coming from the protonated amine group. So that is the carbon thing. In addition, we can also get iron oxide chemistry. As we all know, iron can present in different charge state from 2+, 3+, and combination of 2+, 3+, like in Fe3O4, or in rare cases, it can be also in 4+ in perovskite structures. So by looking at this data, XPS data, you can clearly see there is a big difference between each spectrum, right? Here you have a satellite coming closer to the main peak. Here the satellites coming, away from the main peak, and then some of the satellites have a larger intensity, less intensity. So by looking at these kind of spectral analysis, we can identify what kind of iron oxide chemistry or what kind of iron oxide is present in your sample, whether it is in a reduction condition or oxidation condition and things like that. So now I will give 2 quick examples of what we can do in actual sample. So in the first example, we collected 5 samples or 4 samples from a aeronaut forest in New York at different depth at surface, and then different depth here shown here, 5, 10, and 25 centimeter depth. And then we did the XPS on these 4 samples. And here is the carbon to nitrogen ratio, carbon to nitrogen quantification, and then carbon to nitrogen ratio. You can clearly see the surface have more carbon compared to the carbon deep into the soil. Similar, we observe similar trend is observed in nitrogen too. So here is a nitrogen spectrum from 4 different samples here. So you can clearly see the nitrogen at the surface have a larger peak compared to the nitrogen in the deeper into the soil. And also you can see the shape of the nitrogen peak is different in the deeper soil than in the main soil. So if you compare quick deconvolution analysis, you can see here, this is the nitrogen spectrum from the RT1, and here is the spectrum for RT2. You can clearly see they are different. And then to fit this, we have to add additional nitrogen peak, which is the oxidized nitrogen. And then here is the other 2 nitrogen species. And then from this, you can get some conclusion that in surface region that you have more amine. And then if you go deep into the soil, you are getting more proteinated amine and then oxidized amine. So this is one example. And then we can also, Ajay will talk about this more here. So this is the conventional way of analyzing that sample. We put different peaks here and knowing our knowledge. But we can also do differently like this shown here. So what we did here, we ran several standards which contain these kind of compounds, and then we generated the database of this nitrogen, and then we try to fit this. We try to fit our data using linear combination of these peaks here. So by doing that, we use 2 components from plant derived nitrogen and then 2 components from the microbial derivative nitrogen to fit this data properly. So this is, we just put it randomly like this, but in more, Ajay will explain how we can do AI to do fitting like this. The second example, we separated the whole soil into 2 different fractions. One is above 53 micrometer, and the next one is less than 53 micrometer. And less than this fraction, we mainly have mineral associate organic matter. So when they filtered, they found 2 different kind of mineral assisted organic matter. One is a light color one and the heavy color one, and then we did XPS and then some mineralogy on these 2 samples. So here is the surface composition. As I mentioned, you can use XPS to calculate the surface composition. You can clearly see from this equation, from this figure, that iron and magnesium is enriched at the surface, or the light mineral assisted organic matter compared to the heavy one. We can also get some carbon speciation. You can again see the carbon species is completely different in both from the light versus the heavy. And from using this equation, we believe that the carbon chemistry is more microbial derived carbon than the plant derived carbon. So this is just a 2 examples. Now we will go through why we need the high throughput analysis. So as we all know, the EMSL is leading a big campaign, soil campaign called Molecular Observation Network, or MONE. So the aim of this campaign is to create continental scale database of molecular and molecular structure data to enhance understanding and prediction of soil carbon and aerosol process. So to do this, we need to get some information about the surface carbon composition, both organic and inorganic. So what do we know so far from this MONET mission? They already collected so many samples and so many different regions. So what we know about the carbon, they measure total carbon using some combustion method, but unfortunately that measurement cannot differentiate between organic carbon and inorganic carbon. To get some information about the inorganic carbon, they did some water extraction and then did some FDICR mass spec, and that will give some loosely bounded organic carbon and some different fraction. So what is missing is the organic carbon that is tightly bounded to the mineral surface. So that's what the XPS will play a big role, because as I mentioned, XPS is a powerful technique to get the organic composition of the soil surfaces. So studying XPS on these samples will give a carbon speciation in the unaltered sample surface. Okay, so this is the traditional way of mounting. Here I am showing 3 traditional way of mounting soil sample for the XPS analysis. So in the first one, we basically put the soil on the plate and then covered the soil sample with another plate. And here we put the soil particle on double-sided carbon tape and then press it hardly and then do the XPS. And here you put the soil particle on the silicon wafer and then do the analysis. So the problem with this analysis is you can see the only few samples we can put it here, and then we cannot use the same sample to do some other analysis like SEM or Raman or other experiment. So we need to have a rapid sample preparation method to prepare the samples fastly, and then also to easily transport between instrument to instrument to do different analysis. So we use dot plot instrument to analyze this sample, to synthesize this sample. So basically this plate, this instrument has two plates. Top plate as shown here, it has several holes. The second plate also have a hole, but it is connected to the vacuum pump. We put a filter in between these two plates and tightly connected these two plates and then start pumping it. And then we extracted the soil sample, mixing with the DI water, and then took some 50 microliter of the suspended soil, and then we put it into this hole. So while we putting it in the soil, into this solution into this hole, the vacuum pump suck the water out and then leave the soil residue at the filter. So these soil residues are embedded very nicely so that we can move to the different we can move this filter paper for different instrument. Here is the soil particle in one spot, so you can clearly see we have a uniform amount of soil present in that sample. So this is the workflow for our experiment. So we can do all kind of multimodal analysis, and then finally we can do some AI analysis. So since Catherine is going to talk about using this sample preparation technique into the studying using the XPS to study the soil samples, so I will forward that to her. So basically what I'm going to show here is as an initial testing, when we test up this measurement, we had 41 samples collected from different sites, and then we did some topsoil and bottom soil, and then you can clearly see there is a carbon speciation is different versus topsoil and bottom soil in some samples, and then in some samples we didn't see any differences. So I will skip this one. So basically, the conclusion from my part of the talk is we can use XPS to get nice carbon, nitrogen, and ion speciation in soil samples and in a normal way. And we can also develop a high-throughput method that we can synthesize large amount of sample that can be transferred between different techniques. And then Catherine will show some data from this methodology, how she used our newly developed technique to understand some of her questions. So it's now over to Catherine.
Okay. All right, looks like I have some controls. All right, hi. Yes, like Shuttha said, I'm Catherine Pettinger. I am from the University of Wisconsin-Madison, and over this spring semester, I've been doing an internship here at EMSL, mainly looking at XPS as well as some other methods, and to better understand some soil processes, kind of all the ones that Shuttha was talking about. If you're on this call, you probably know that soil structure and composition is obviously very influential for many processes, everything from carbon cycling to soil health to how contaminants, nutrients, and water move through the soil and the subsurface. And one of the big interests recently has been especially in how do we model carbon fluxes and look at thermodynamics from soils, and what are some different tools to do so. And especially here at the EMSL lab, one of the ways that has been done is to look through the Monet project. And for those of you familiar with the Monet project, you probably know this already, but basically they're collecting samples from across the continental United States. And they've been expanding that more and more to cover more soil orders, more land uses, and collecting standardized data sets ranging from metagenomics all the way to how much water is in soil. And with all this different data, we can start to really put together a picture of what is happening in the subsurface. One of my questions that I've been looking at while here is how do we also fit in some soil mineralogy, such as bulk and surface, compared to already collected data types, and what drives any of these variations that we're starting to see. So to do this, we started with 25 sites. That covers 11 of 12 soil orders. Each of these sites, there's two different layers that we're looking at. One is the top 10 centimeters, and then we're also looking at the 20 to 30 centimeter depth range. And I'm looking at XPS data. I'm starting to look at some XRD and SCM EDX data. And we're going to, just for today, talk about the XPS, but more to come. Okay, so she'd already talked about how we're prepping the samples. We were able to run 50 samples in our first run, which was amazing. We did have a little bit of soils that leaked into each other. Basically, I pipetted too fast, and some of the water kind of leaked between them, which also carried some soil particles. So we repeated those samples on a new filter, and any of the other ones that also had lower yield of soil on the filter. Okay, so she just talked about the survey spectra. So I just kind of want to show what those results look like when we're looking at many soils. And one of the things we can get is this relative abundance of the atomic concentration. So this is looking at how much silica is there to oxygen, to carbon, to aluminum. And keep in mind, a lot of this oxygen is tied up in like silicon dioxides. And we can then get the overall percentage. We can also look at, specific individual elements just by taking each of those separately. And we can start to compare them to the bulk values. So from the Monet data that's already collected, we have bulk values of things like carbon, nitrogen, the major cations and anions, etc. So just looking at carbon, for example, we see a lot more surface level amounts of carbon. This is going to be carbon that is a little bit easier to access typically by for soil respiration, microbes, etc. So it was so this kind of matches what we expected as well, that there'd be more carbon at the surface of a soil particle than in the full grain. And then we see that also for nitrogen for most of the soil orders. However, we do see, for example, the vertisols, there's a little bit more bulk nitrogen than what we saw in the XPS for that sample. But we're starting to get a good idea of kind of this might be influencing carbon nitrogen ratios at the surface that is active compared to looking at carbon nitrogen ratios in the bulk to better get a holistic picture of what's happening in each soil particle. So then Shuttha also talked about some high resolution scans. So this is what two of them look like for some of the sample site CLBJ. As we can see, the top has more oxidized carbon formats. So again, those oxidation functional groups will shift some of the carbon peaks to an increased binding energy compared to our lower level sample. This isn't true for every sample, but how much of this happened varied between each sample. But I was kind of interested in these peaks because it reminded me of the FTICR data that's been collected, where we also see kind of this oxidation state curve. So I wanted to see if these match, if they're similar, and long story short, they're not. So the NOSC value calculated from FTICR is an estimate based on many different organic molecules detected. There's lots of information out there on how to interpret FTICR data that you can find, so I'm not going to go into depth about that. But basically, the data types don't match well, and this is partially just due to how we look at carbon oxidation states. So in XPS, we're looking at increasing the oxidation state to above 0 with additions of electronegative groups. However, carbons range from -4 to +4, and carbon hydrogen groups or carbon bound to hydrogen will decrease the carbon oxidation state based on classically assigned carbon oxidation species. So this is just important to keep in mind when trying to compare data types, is that we're lumping together the carbon-carbon-carbon hydrogen bonds due to their similarities in binding energy. The pro of what we're looking at is that we can really start to see these functional groups that are looking at, oxygen additions and other electronegative elements that can increase the oxidation of carbon beyond 0. Okay, so I did want to just try to adjust the FTICR oxidation state scores, even though they're calculated very differently, just to reflect the carbon, to ignore kind of carbon-hydrogen bonds. Some of them start to match, some of them still are not. But again, this is a little bit because of how the data is collected, what extractions are performed. The FTICR is using a water extraction. But one thing I did want to point out is when we look at these blue boxes here. So on this, I'm looking at the top. So this is the top 10 centimeters versus the bottom layer. So that's the 20 to 30 centimeter core depth. And we have our oxidation states on the y-axis. And we see that the bottom samples are generally have a lower oxidation state than the top samples. Again, this is as expected. And this also might be indicating that our lower level samples are having more of those carbon-carbon, carbon-hydrogen bonds, which would be indicative of more like lignin-like structures or lipid, which are also less soluble, so which might explain kind of the discrepancy between some of the FTICR and the XPS data. Okay, so one of the big things people look at when doing soil respiration modeling and other metrics for how active different elements are in the soil is just how much carbon there is. So I wanted to see if that XPS carbon oxidation state that I basically averaged out matches or is correlated at all with the bulk weight percent. And we see a very weak correlation. It's not incredibly strong, which environmental data can be like that. However, one thing that I did note is that when we start looking at the individual soil orders, so taking the subset of samples that have similar soil properties, and we start to see for some of these, we have a tighter correlation. Not for every soil sample type, though. And then even one of them, we had like this R-squared of 0.97. Again, this is just based off of 6 data points, but still is really interesting that we have like some really closely tied oxidation state to bulk carbon values that seems to be soil type dependent on how closely that relationship is. Okay, so again, carbon is related in a lot of models as being one of the bigger predictors of soil respiration, such as in this paper. So I also wanted to see just how does our bulk carbon compare to respiration rates. This is another data type that is available on the Monet webpage. If you're interested in how that exactly is determined, I recommend you check out some of that Monet data sets. But we see a pretty tight linear correlation for a lot of the samples. We obviously have a few that are being a little bit exceptional, but we have a pretty nice correlation for a lot of them. So I was kind of wanted to hypothesize that if we did see this correlation between our oxidation state and our bulk carbon, we also see a correlation between our bulk carbon and respiration. Do we then see a correlation between oxidized carbon to high respiration? And again, there's a lot of confounding factors here, but just to see in a quick linear correlation. And this also would make sense in that with more functional groups that are electronegative, that would create some dipole moments that would be a little bit easier for another molecule to come in here and break off these carbon dioxide groups. Okay. So to plot respiration as a function of the XPS determined oxidation state, we see that it kind of is fairly low until you hit this 0.75. And then some of our samples really take off, some stay lower, but we don't see a lot of increased soil respiration unless you get to this oxidation state of 0.75. I'm still working out why that might be, or if there's other factors that are more important. But again, just this is how I'm calculating that carbon oxidation state is looking at the area times these different oxidation states, averaging that across the carbon. So higher oxidation states, more electronegative groups, potentially, depending on maybe other factors involved, higher respiration. Okay. So lots of preliminary work. I'm happy to hear suggestions. As both a grad student and an intern, my main job is to learn. So if you have any suggestions moving forward, feel free to contact me. Right now, what I'm also looking at is incorporating some XRD data, especially as we start to look at different minerals that might be involved, if any of them are corresponding to different carbons that are present. Also, SEM, EDX. The SEM was really useful to look at the sample and make sure we had sample coverage for when we did the XPS run. This kind of helped verify that we're truly getting the soil and not the filter paper. And then we can also do EDX analysis to kind of match how the silica, aluminum, iron ratios are different in that very tip of the surface versus the near surface by EDX. And then looking at some metagenomics, I have some hypotheses relating to carbon cycling genes and how, or if they're correlated with some of these XPS oxidation states, as well as if the metagenomics are more tightly matched to some of the FTACR data. Okay, so again, thank you for EMSL for hosting me for the last few months, and NSF for helping pay my stipend and supporting this project, as well as my lab back in Wisconsin-Madison. I'll be back shortly.
And our next and final speaker is Ajay Karakoti. So Ajay, are you able to come on? Ajay, you are still on mute.
Can you hear me now?
Yes, thank you.
Okay, and I do have the control. Okay, all right. Well, good afternoon to everyone, or good evening, depending upon where you are located in the country. First of all, thank you to Shuttha and Catherine for giving that wonderful talk and how we can use XPS for soil analysis. What I'm going to talk to you today about is what we are trying to do in future and hopefully present it to the users of integrating AI and ML-based techniques in XPS data analysis. And before I begin that, let me also put a disclaimer that I'm not a data analyst. I'm a material scientist and a very heavy user of XPS. So this has spurred from our need for correct interpretation of XPS data. So we are collaborating with multiple scientists over here at PNNL who are expert in AI and ML to help us with the XPS data analysis. Okay, so as you have seen, Shuttha has shown you multiple things that XPS can do. In addition to just the component analysis, as in how much of carbon, nitrogen, oxygen is there. So we just don't want to do composition analysis. We also want to know oxidation states of different materials, what are the bonding states, in which case it becomes very complicated very quickly. So XPS data analysis as of now is very heavy user input driven. The complex spectra that you see on the screen is an example of a sulfur scan, high resolution sulfur scan, and you can see that there are multiple peaks that are fitted to this particular sulfur scan, and each of these have got a different bonding environment, right? So the accuracy of XPS peak fitting is generally dependent on how much user has experience with these things. And the inaccurate XPS peak fitting is very, very common to find in research publications. Moreover, when you start doing the fitting, and especially when you're looking at thousands of samples or maybe even 50 samples of XPS, And when you're thinking about 50 samples, but if you're looking at the 6 different components, that quickly becomes 300 different peaks that you have to fit, or different elements that you have to do peak fitting for, and you have to manually constrain areas and identify peak positions and do sort of full width half max analysis and a lot of other things. So it becomes very, very complex, right? So even though XPS has become a very common tool for surface analysis, if you go into the literature, it is ripe with incorrect XPS analysis. So for example, over here, you see people have tried to fit different magnesium peaks here and ascribe them to different types of manganese oxidation states. If you show this to any XPS expert, I mean, there's nothing more than noise over here. It's very hard to really see anything. But it is very commonly found in literature. There are examples of incorrect peak fitting, wrong background selection, fits not even following the actual analysis. And recently, we have also started seeing people just completely ignoring the physics and doing inaccurate peak assignments. So for example, in this case, you can see there is a spin orbit splitting of SP to be 3/2 and 1/2, and they are in a particular ratio, 2:1. Whereas when people start fitting these peaks, they just completely forget about the spin orbit splitting and their ratios, and they start fitting to their beliefs, what they expect from the sample, rather than what is the data saying and what is the underlying physics. So we are aware of these issues, and obviously these issues will sort of magnify as you start interpreting a larger and larger amount of data. So how to improve the XPS data fitting? If you go and find out in the literature, people have started attempting this. There are multiple reports on using machine learning models to improve the data analysis for XPS. We are taking a very similar approach, except that most of the machine learning models these days that are out there have used only theoretical data, and many of them have also only restricted to the elemental analysis. Whereas what we want to do is we want to use an automated Python-based course for running these scripts so that you can do all of these automatically. It picks the background, it assigns the peaks, and it constrains the peaks so that you get very accurate fitting results. What we are doing in the background here is we are trying out all these automated scripts and comparing them with the manual fitting that we would do, and trying to improve our code and our algorithms that we are using for fitting these. And one of the things is obviously physics-based full width half max constraints that we are trying to put into the system that allows you to fit these peaks better. And then hopefully in future, what we want to do is combine this code that we are using for this fitting with what Shuttha had said about pattern recognition. So there's always some use of already a use of large language model informed XPS data analysis in literature, and here's an example. But what we want to do is we want to sort of combine these two things together. And hopefully present this sort of a automated workflow to an EMSL user, where there will be an XPS data which will be automatically fitted, and from which the model will derive the peak parameters and then feed it to large language model based analysis of these peaks. And this is where some of the things that we are trying to do. We are not trying to go directly into the literature to compare. We are still relying on what literature is to be fed to these large language models, or even a background ML algorithm that looks at a very specific training data set, which is not just a theoretical data set, but also a combination of theoretical and experimental data set. And then identify those chemical states, bring it to the expert, and then continue to do the data refinement. What we are also doing in the background, and this is what Shuttha was referring to, is that instead of giving XPS, which is standardly used as a tool for oxidation state analysis or binding environment analysis, which may or may not be very useful for a user who is not aware of this. So for example, over here, there is a nitrogen spectrum which says there are amine type carbons, there are primary amines, there are protonated amines, and there is some oxidized nitrogen, right? But it doesn't tell me whether it is belonging to a protein class, whether it is belonging to some nucleic acid, whether it is a nitrogen coming from chitin. So what we also want to include is some sort of a pattern recognition, so that when we present it to a user, we start to fit these XPS spectrum to a particular pattern, and machine learning models are very, very good with this pattern recognition. So we want to do pattern recognition and then merge it with the XPS peak fit, and then try to improve those models to accurately define not just what is the oxidation state of these components within this particular sample, but also what would it ascribe to. Of course, we understand it would also depend upon some user information, some background about the sample, and that has to be fed beforehand before we start analyzing these data. So overall, our hope is that we would come up with some sort of a model. This is what we are trying right now, where we are starting with XPS data set. We are doing some statistical fitting. We feed into a primary LLM, and then this is where the most of the work is going on, insights with reasoning, where we are trying to combine pattern recognition, machine learning, and training. And then finally, we will hopefully go on to judging with different LLM models, and then that would refine the prompt. We could give these prompt to the user depending upon what they want to see in their data, and then finally there has to be an expert in the loop. So this is the work in progress. We are hoping that we would soon be able to present it to our users, and that would probably help them with the correct interpretation and correct analysis of the XPS. And that's all I have for you guys. Thank you.
All right, thank you, Ajay. So we are going to move on to the Q&A portion of this webinar. So again, folks, if you have questions for our speakers, feel free to put them into the Q&A portion or into the chat, whichever. So let's see. Let's start with Kevin had a question. And this is for Shuttha. How is XPS different than XAS? They seem like similar techniques. I'm interested in XPS to differentiate types of mineral phosphates like calcium phosphates, iron phosphates, aluminum. Would XPS be able to do this?
Yes, so the XAS is, from the beamline, if I am right. So that is actually probing a little deeper than the XPS depth information. So we are really collecting at the top few layers of the material, like 3 to 10 nanometers. So that is the first difference. And definitely, we can differentiate with the different phosphate if we can see a large difference in the binding energy. Okay, so I'm not sure exactly how much is the difference is going to be, but definitely we should be able to differentiate between different phosphate, looking at the different, at least looking at the phosphate signal as well as looking at the aluminum and the calcium signal.
Okay. So the next question is from Trang, and this one again is for you, Shuttha. Could you please give us a sense about the time and cost estimation for each soil sample using XPS?
That, I don't know, this is a user, the time one is, see, the normal way of doing it, it will take some time because you have to put 4 samples at a time, right? But the way that we develop that, the idea is like we can analyze, we can collect the data, 50 sample in one day, like carbon. Mainly, we are targeting carbon, nitrogen, oxygen, and a survey spectrum. That's what in our workflow right now. So that will give us, like in 24 hours, we can collect 50 samples per day, and then maybe analyze will take another 1 day. Catherine can explain how much time she spent, because once you fit it, then you can just propagate all the fitting. So it is pretty fast analysis. So that is the time constraint, right? But if you want to run iron and some other elements, then it will take a little bit more time because the signal is low. The intensity is low, so you have to run more time to get a better statistic. So what I will say, if you want to run both the organic set and the iron and the calcium and phosphorus set, it will take 2 days to run 50 samples, and then maybe another 2-3 days to analyze the data. And the cost-wise, I cannot answer that up to EMSL. You have to put the proposal, and if it is funded, the staff time is and instrument time is covered. So,
All right. Great. So AJ, this one's for you, from Don. He would like to know, with AI ML analysis, will you be able to assess the uncertainty in the results?
Oh, I think Don already knows this is a hard question to answer. What we do want to do is we want to address the uncertainty in results. One of the goals would be, I mean, there are two levels of uncertainty, right? So there is uncertainty in the spectral data that we collect, and the noise and the fitting to that noise and stuff like that. And on top of that, there is an uncertainty that can creep in based on what our model is doing, right? If both are performing at the worst end, then your uncertainty actually multiplies. But if both are performing at their best end, probably the uncertainty can be reduced to an extent. But what we do want to build into our models is that we have to sort of build in that, okay, this much minimum signal to noise ratio should be present before we start fitting the data, right? So that takes out some of the things. Then we can also, these days there are neural networks which are uncertainty aware neural networks. So probably we will have to involve those uncertainty aware neural networks into the fitting analysis. And that's how we would probably able to reduce the uncertainty. And as I said, it is all a work in progress. So that's why there is still, for a very long time, in the given future or foreseen future, I'm hoping we will have an expert in the loop who would continue to validate these analyses by the models. And the whole goal is to avoid very basic mistakes in XPS peak fitting, and also to provide a detailed analysis which is sort of physics based rather than intention based or whatever they want to see in their sample.
All right. I'm not quite sure which one this would go to, possibly Shuttha. So this question, how reproducible are spectra from each soil sample?
yes, so that's a very good question. So we did, first of all, it is not uniform, right? So each soil, even within the region, will be different, right? But what we did several different runs, as Catherine pointed out. Initially, we started in her sample. Some of them didn't work well, and then we had to repeat it. And then we put some other samples also that worked very well, and we were able to reproduce the data within, our experimental uncertainty. So it is pretty good that we can reproduce the data in two different runs in using our newly developed technique.
Right, and if may I add,
yeah, sure.
It also depends upon how much area we are analyzing as well. So right now, if you have a large area that you can analyze within your given sample, and that large area is sort of representative of the whole sample, then the chances of it varying from spot to spot are minimal. So it also depends upon, obviously, your sample. If the sample is very heterogeneous, then the data would also probably be heterogeneous from spot to spot. But if we use a larger spot size for analysis, then we can basically look at few hundreds of microns of the area, as opposed to what you would do in a SEM where you're looking at maybe 25 micron square area. So we can look at few hundreds of micron square area with our XPS, and then that's why it is more representative.
Yeah, so that's what the main goal of this this work, that technique that we developed, to run XPS and do SEM and other techniques in the same sample spot. So it will be, we can compare apples versus apples rather than apples versus oranges. So we have some data. Catherine collected some beautiful SEM data. Unfortunately, we cannot show everything. But if anyone wants to if you want to see those data, please contact us. We are willing to show that data. We have some nice. We don't know whether we can really correlate the XPS data with the ICM yet, because it is just we finished that project. So, but I think pretty sure, we should be able to get some very useful information combining those two measurements.
Great. The next question is for you, Catherine. What factors of soil orders do you anticipate being important to how well oxidized carbon content is to total carbon concentration or soil respiration rates?
So there's lots of factors that go into what makes a soil order a soil order, including the depth of the different soil horizons. So especially when we start to think about like how different that like top 10 centimeters is from the 20 to 30 centimeters, depending on what soil horizons are present, which will vary by soil order. We can also start to think about like water content or iridosols had a little bit less variation in like the different layers compared to a more highly moist soil. And so like that wet factor can play a big difference, as well as temperature and what the original parent bedrock material is that formed the soil. So lots of possible factors that can all work together to change what we see in the soil.
Okay. Excellent. Let's see. Can we use XPS to measure the carbon stock in the soil?
Carbon stock.
As
I
in
think so.
total
I mean,
carbon?
stock?
Yeah, can we use XPS to measure the carbon stock in the soil? I guess, yeah, total carbon would be my, yeah.
Probably, yeah. I mean, at the surface region, that's what we are doing, right? The total amount of. So I don't really understand what is stock mean. I mean, if it is just a carbon bond, that is the tallest peak that we saw it in our spectrum, in some of the spectrum, right?
I think I'm looking at the Q&A, and then the person who has asked the question is asking the total carbon content. So like
okay.
the
So
total
yeah.
carbon that we get from
Okay, yeah, from the surface, yes, definitely, yes. That's what Catherine was comparing, right? The total surface carbon obtained from the XPS versus the total carbon obtained from the combustion measured. So you can clearly see there is a big difference between the total carbon measured from Monet versus the surface, the total carbon versus the surface carbon measured from XPS. So always surface carbon shows higher value compared to the bulk carbon values.
Mm-hmm.
Because XPS is a quantitative measurement, so.
Okay, we have another one as well, another question. It was a couple of them. Is the rapid sample method available for EMSL users right now? Like, is it ready in this moment?
Yes.
Yes. Awesome.
That's what Catherine did.
Catherine already showed that one, right?
Yeah, no, the person joined late, sorry.
So yes, you can request it, anonymous attendee. And then anonymous attendee also is asking, how does the run rate differ for the high resolution mode versus the survey scan?
Oh, yeah. So that's again a good question. So it varies, depends on, again, how much each element is present in that sample. See, sometime, if you have a lot of carbon, then we don't need to run high resolution that fast. So basically, rule of thumb is, for the low resolution mode, we run, say, 5 scans. And then high resolution, we run like 20, 25 scans with a different analyzer condition. So that's the reason that, when you run in the high resolution mode, the intensity reduced because we are reducing we are increasing the energy resolution to separate those peaks. So we want to have a nice separated peak so that we can fit nicely, So the high resolution can take long time, definitely. But the surface scan is just a quick scan just to identify what are the elements present in the surface, and then we go into each individual element and then we scan them in a slow manner. So probably 1 to 10, 15 times difference.
Great. And with that, we are out of time. So I'd like to thank our presenters, Ajay, Catherine, Shuttha. Thank you so much for presenting today about XPS and the possibilities with soil analysis. Thank you to all the attendees for joining us. We hope to see you back here next in 2 weeks. And our next webinar is on May 28th, focused on vibrational some frequency generation spectroscopy. So another one that it's another capability that can help with your critical minerals, metals, and other biological and environmental research applications. So again, thank you, everyone. We hope to see you in a couple weeks.